right. The sound's okay? I don't need to wear a mic or anything. Okay. All right. Well, so happy to kick off this conference. I've never attended this RECOM Regulatory Systems Genomics, but every year I look at it with admiration and curiosity and interest. So I'm excited to get to participate and extremely excited to get to lead it off here with the presentation. So I'm going to give you, I think there's a couple entries into this type of work. There's a couple of reasons, there's many reasons to do this type of work. The one that is most compelling to us at Calico is through human genetics, but I'll try to note for those in the audience who don't care that much about genetics, some other reasons as I go. So, for us, we're very interested in common diseases. Calico is interested in aging. Aging is, you might argue, the most common of diseases. It's going to happen to all of us. And all of these common aging-related diseases now have genome-wide association studies from human populations that have highlighted many, we're talking hundreds and hundreds in some cases. This is EGFR, marker of chronic kidney disease, and you have over 800 associations from a GWAS, each of which is a whiff of what's going on in that disease. And at this point, these are very optimistically labeled with genes. In some cases, the genetic variants are hitting coding sequence or introducing a stop codon or something that we really understand well, and you can confidently label that gene. But a lot of them are optimistic. They look like this, maybe. There's a lot of ambiguity about what the causal variant is because of linkage disequilibrium between variants. Basically, like the variants are very correlated in the population, so you don't know which is the real one. And generally, there are multiple genes in the locus that could be driving the association. So for us and for many in the field, this represents, I think, an amazing opportunity to learn about human biology of disease and of any sort of trait that you can go out and phenotype in a large population. But you really need to add these additional objectives onto having just done your GWAS, which is to precisely identify those causal variants and determine how they affect phenotypes. So what we're learning from a lot of these studies is most of these associations are non-coding, not very convenient, but again, representing a great opportunity for those of us who really like to think about how the biology of gene regulation works as a function of sequence, because it means we can kind of enter this field and try to be useful. So that's kind of where I'm at now, entering this field, trying to be useful. And for these non-coding associations, the objectives that I want to lay out are highlighted there at the bottom. We want to know the causal variants, but we also want to know which gene is the affected gene that's really mediating this association signal. And then now that we have profiles that represent lots of different cell types and states, we really want to know which of those is most relevant to mediating that association. Okay, so the approach to this problem has been to fit these models that take in DNA sequence as input and try to predict properties of gene regulation as the output. And as Farhad highlighted, we've published a few papers in a series trying to work up to better and better solutions to this problem. The latest one that I'm depicting here is our informer method, which apparently everyone has already heard of. But if you haven't, it's basically a deep convolutional network where the initial layers of the network are these convolutions. They can capture things like transcription factor binding sites and other sorts of relevant motifs. The subsequent layers of the network can start to stitch together more complicated patterns, recognizing enhancers, promoters, insulators, whatever else is out there. And then in this work, the kind of novel contribution was to take advantage of this deep learning block called either the transformer or what I like to call it self-attention, where you basically have a mechanism for any two positions which are represented as vectors after having processed through multiple layers of convolution. Any two positions can communicate in the network through these self-attention layers, where there's this mechanism for saying, I'm looking for this, I represent this, there's a attention matrix that's formed, and some vector propagation of information. Sorry, I'm being vague, but I want to get to some of the more interesting stuff. And transformers are very Googleable now if you want a much better tutorial than I can deliver, because they're the core component of these language models that are slowly taking over our lives. So on the right here is a depiction of just the general framework of what we're trying to do. We've got the genome data in green. So the way we're going to set this up is we're going to train on most of the genome. We're going to hold out a smaller portion of the genome, and that's going to be our way to evaluate how well we're doing and decide when to stop training and so on and so forth. So this is a test set sequence. The model hasn't seen this sequence, and you can see that qualitatively, the informer model is exceeding the performance of the previous model, Basenji, which the main difference is it doesn't use these self-attention layers. So self-attention is an exciting development in deep learning, and we wanted to take it forward and see what else we could do. Every time I've given this talk over the last few years, I try to avoid talking about RNA-seq because I recognize that RNA-seq is the most common functional genomics assay, and yet these models previously, Basenji and Former, are not predicting RNA-seq. And the main reason is just that I thought RNA-seq was too complicated. Basically because you have these spliced aligned reads, you could have a very high, these models are they're very naive. They're just taking DNA sequence and they're trying to predict this aligned coverage track in bins across the genome. So if you picture a really, really long gene that's highly expressed and it's 3' UTR, the promoter information and potentially enhancer information that helps you determine how many reads are going to align to this 3' UTR are really far away. So there was no way that was going to work with the dilated convolutions. It's only with the self-attention that it really became possible, but I didn't quite realize it was possible for a little while. But at some point we did, and let me kind of justify why you would really want to go for this objective of predicting RNA sequencing. First, like I mentioned, it's the most common assay. If you go look in Geo, I'm sure there's more RNA-seq than anything else in there. At this point, most of our single-cell experiments are using RNA-seq. There's lots of single-cell attack-seq and chip-seqs, and that's all great. But if we're going to profile some really rare cell state, if I had one tissue sample from one person, I'm probably going to go do an RNA-seq. A lot of our assays that profile specific aspects of post-transcriptional regulation, here I'm thinking of RNA binding protein binding with ClipSeq, different assays to measure RNA stability, like a half-life measurement time series, the RNA methylation marks like M6A, as well as different secondary structure profiling methods. They're all basically glorified RNA-seq experiments. There's a lot of clever steps to focus on the thing you want to focus on, but then you do an RNA-seq. So if we had models that could work with RNA-seq, it would give us an opportunity to tap into all of these things. There are machine learning models as a function of sequence in this vein of Informer for different specific aspects of post-transcriptional regulation, such as splicing, polyadenylation, and some of these other processes. And they work great. You've probably heard of Splice AI. That's a very effective program for predicting splice sites. But there's some limitations when you really look at these and ask them to do more complicated things. They're isolating a process. They're working with training data that's highly processed versions of RNA-seq experiments with lots of other information. So it's a little bit unsatisfying. You'd like to be able to get a little closer to the data. And then finally, there are a lot of interactions between these regulatory layers. Probably the most well-known is that the splicing machinery will engage as transcription is happening. So there's co-transcriptional splicing. And so there's lots of different opportunities for a model that can unify all these different layers of regulation to make smarter predictions and to help us better understand these interactions between the layers. So that was our objective. we're basically tweaking the informer architecture and building in a few new tricks that we've learned over the years in order to predict RNA-seq data. So there are a couple things that I really wanted to modify from the informer architecture. One was I thought it was important to decrease the resolution of the data, the output bin size that we're predicting at. So with informer, we're predicting at 128 base pair resolution. Like to think of as like roughly, the distance between two nucleosomes if I was stuck with powers of 2. And that seems fine for a lot of ChIP-seqs. You look at your peaks and the blobbiness of it is about 100 base pair blobbiness. And for something like a TAC-seq, if you're thinking at just a peak level, that's probably fine too, although there's now some really great work looking at finer resolution cut sites from a TAC-seq. So basically, finer resolution seems to be better. And for RNA-seq, it's particularly important because there are exons that are quite small, some of the internal exons of these genes. And if they're smaller than the bin size, then it gets kind of weird to think about what's actually happening there. So we wanted to zoom in relative to informer. The problem is what you'd like to do is just convolution to the size that you want, and then do your self-attention and then do your prediction. That's the kind of informer architecture. The problem is that we also wanted to increase the length of the sequence because a lot of genes are really long. They can be hundreds of kilobases. We need them to fit inside the sequence. We can't have cases where the kind of 3' end of the gene is hanging off and all the transcriptional information is off the sequence and invisible to the model. So, okay. We want to increase the sequence length. We want to decrease the bin size. The problem there is that the self-attention has this quadratic complexity because every 2 pairs of positions in the sequence are getting compared. And that blows up the memory of our GPU RAM. These GPUs do not have a huge amount of RAM. The most sophisticated ones have the A100s and now H100s can have 40 gigs of RAM. So that's kind of our limitation. Those are also now very hard to get, again, because of language models, which are taking over the world. But nevertheless, we couldn't do self-attention at the sequence length that we wanted to do. So instead, we're borrowing a technique that's become very popular in image analysis, which is called a UNet architecture. And the idea is you want to be kind of processing your image at a low level and then doing pooling operations and kind of decreasing the resolution of things until you can get a kind of global view of what's going on in the image. But then you want to zoom back down to the pixel level and actually label what's going on in particular pixels. I think if you're doing something like segmentation of cells in a microscopy image, you very well might want to take advantage of something like this UNet architecture. So for us, this basically means that we're continuing to do convolutions and pooling on the left side here, but then we're going to do our self-attention / transformer blocks, and then we're going to go from 128 base pairs back down to 32 base pairs by passing the representations across and doing a sort of combination neural network block. You just you do a transformation of this and a transformation of that, and then you add them together with these residual connections, and it all kind of works out. So those are the main insights into the new architecture. There's a few optimization tweaks to make it all work really well, but that's the general idea. Okay, so does this work? Roughly, yes, it does. Here's an example of a moderately complicated gene, again from the test set, where the first row represents the RNA-seq coverage track, the real data, and the second track represents our model, which we named Borzoi, by the way, our model prediction for this gene. So you can kind of qualitatively see that we're picking out all of the many, many exons of this complicated gene. At the bottom, I'm giving you predictions and the data aggregated now to the gene level and computing an accuracy of being able to predict gene expression from this new model. On the left, it's just kind of predicting the raw values, and then the histogram is filled with all of the tracks that we trained on. So I should say the training data tracks for this model are all of the ENCODE RNA-seq. We do that because the ENCODE data is readily available and very easy to access, and we appreciate that. And then a lot of data that we accumulated from the ReCount database, which is another incredible resource of uniformly processed RNA-seq data. In that case, we're mostly grabbing the GTEx data. So on the left, everything forming that histogram is one data set, so you can kind of see the distributions of the accuracy levels for all the different data sets. On the right, we're trying to take on this harder task of saying that, well, there's lots of genes that are more or less expressed at the same level across all the tissues that we see. Those tend to have less complicated regulation, more promoter-driven. They're a little easier. So let's normalize all the data sets carefully against each other and then subtract the mean. And then you have to predict the residual of what's left, the differences across all the tissues. And even then, we're pretty good. So there's definitely some tissue-specific information that is learned. I often get the question, how does it learn tissue-specific information when you're giving it the same DNA sequence in every case where you're trying to predict all these different tissues? And the general idea is that it learns the transcription factor binding sites that are most active in that tissue, and it differentially uses them to make predictions in those different tissues. So to show that to you, I've got 5 GTEx tissues here. You want to start in the upper left of this semi-complicated figure and see that what we did was pick out here a bunch of genes that are differentially expressed for each of these 5 GTEx tissues. The one you're seeing there is a liver differentially expressed gene. And then what we're doing is we're taking the gradient of the model predictions aggregated on top of the gene. So all the bins that overlap the gene's exons, sum them up, and then take the gradient of that with respect to the DNA sequence. So what you're asking is, if I take this nucleotide, which happens to represent an A, and I make it less of an A, then what's going to be the change in the model prediction? This is kind of an approximation of actually introducing the mutation, which is something we also like to do a lot. But in this case of wanting to score the nucleotides around a gene, which could be hundreds of thousands of nucleotides, it's too computationally intensive to go one by one and introduce all 3 alternative alleles for that many nucleotides. Whereas the gradients you can get in one backpropagation pass through the network. You just have to do it once for each of these tissues. So we had to do 5 back passes to get those 5 little tracks. And that's tractable, especially if we live on the GPU to do it. So what you really want to notice there is that there's nothing happening in the tracks from the other tissues. And that's because this gene isn't really expressed there. The model is recognizing that. And the nucleotides that it's seeing, it only has positive gradients for liver. And you can pull out those motifs and see that they are indeed known liver-specific transcription factors. So if we do that for a whole bunch of genes for each of those tissues, and then we pull out those little motifs that pop up and cluster them using a method called TF-MoDisco from Anshul Kandaji's lab, then here's a set of motifs that we come up with for each of those tissues. There's lots of known transcription factors that are relevant in those tissues. And the distributions we thought were kind of interesting to show you. So each of those dots is one of these motif instances, and the score via the gradients for each of the different tissues that we looked at. So there is some interesting activity across tissues. Mostly those motifs are active in one tissue or another, but there are some interesting cases where that's not the case. We pulled out this CEBPA. We're not really sure which one it is, A/B maybe, here as an example, because there's activity in both the blood and the liver for this motif. But what this little offshoot scatter plot is showing you is that each of those dots is one instance. And by looking at the scores assigned to the different instances, we see that it's actually different instances of this motif that are being used in the blood versus the liver. There's not a ton happening in the upper right quadrant, which would mean that it's an instance that's active in both blood and liver. But there's a lot on those bottom right and upper left, showing that it's really specific to one. So, this ability to make tissue-specific predictions, it's not just taking the motifs and propagating that forward alone. It's looking at these motifs in the context of the regulatory elements that they exist in. What are the flanking nucleotides? What are the other motifs that are around? And using that information, and the gradients can be a nice way to read that out. So just to convince you that all of what I'm showing you here is reasonable and legitimate, in the bottom left, we've got a little validation exercise where we're aggregating for each of the tissues the motif score. So we just have now one score for that motif in that tissue. And then because we're working with RNA-seq, we have the opportunity to just look up the gene abundance for that transcription factor. And in some cases, it's ambiguous and we have to merge it together. But for the most part, you see that there's a compelling correlation between the gradients. If you do a pairwise comparison of two tissues, between the gradient difference and the actual transcription factors expression difference. And, that's not always going to be the case. There are lots of examples where the RNA-seq measurement for the TF is not the best proxy for its activity, but it's something that you do tend to see across these tissues, and it's reassuring to see it. Okay, so one of the big arguments that we make for doing this sort of modeling is that we could take advantage of sequence information beyond the promoter. To return back to this GWAS for a moment, it turns out that a lot of the heritability for these GWASs are coming up in enhancer elements that are distal to the genes, potentially because promoter variants have larger effects and some of them get weeded out by natural selection, whereas the enhancers maybe have more subtle effects. You could debate that, but either way, we really want these models to be able to make good predictions for enhancers. And if they were really, really working well, then we could even use this as an engine to query for promoter enhancer interactions in different tissues where all we had was some functional genomics data, like an RNA-seq experiment. So there's some really great data out there that we can use to evaluate the model's ability to link enhancers to promoters, coming through CRISPR experiments where simply you're using the CRISPR machinery, you're targeting it to a specific enhancer in order to block the activity of that enhancer, and then you're reading out gene expression. This is often done in a gene by gene format where you target lots of regulatory elements around a specific gene. We have data like that for on the order of dozens of genes now. And there's also a really neat data set where it was done by Jay Shanduri's lab in a single cell format where you get just a kind of big random bunch of enhancers and genes in the genome from identifying the specific single cells that have the perturbation of interest and then looking up the RNA-seq expression. Okay. So again, we're going to leverage the gradients of the model. These experiments are all done in K562. So we know a lot about K562 enhancers at this point, a little less about other cell types, but we'll get there. So here's a couple genes where we're using the gradient of the RNA-seq prediction for the gene, laying it out for this big region of sequence. And then I'm running a little smoothing operation over that because I don't care so much about specific nucleotides now. I want to score at the level of like a 100-ish, 100 to 500-ish nucleotide bin. And so these tracks are showing you those gradients. There's actually an ensemble of 4 models, so you can kind of subtly see that there's 4 different lines happening there. And then these vertical lines, there are red ones which represent the negatives in the CRISPR experiment, and the green ones which represent the positives from the CRISPR experiment. So you can see for this HBE1 gene, there's a series of enhancers that are on the order of tens of thousands of kb away from the gene that the model picks out really well. And for MYC, we're highlighting that one because there's this really long range enhancer element here on the order of hundreds of thousands of nucleotides away that we do get right, although you would probably argue for MYC that it's kind of sitting in a small pond of false positives too. So this is something that we're still continuing to develop. And I think on the side of the CRISPR experiments too, these are continuing to get better and denoise, and hopefully we'll both converge on great enhancer maps soon. So for the MYC one, I want to highlight, it's kind of interesting. You can then dig in and use these nucleotide interpretation methods to see what's there. And in typical K562 fashion, if you've looked at it, this enhancer element is a GATA element. So it's definitely, the model has learned that K562 likes GATA. At the bottom there, we're computing, typical classification metrics, AU, PRCs for the enhancer elements binned at different distance levels. And you can kind of see a few trends. The predictions are better closer to the gene than further away, but the new model is much better than the informer at this, and the informer can't even do that final bin size. So that's a nice development in our ability to predict these things. You don't see it. You see a kind of smooth drop off, but our accuracy is still much better than random at that distance. Okay, returning to genetic variants, there's quite a few interesting things that we can do with this model in this space. One that we've done before with Informer is focus on eQTLs, and here's just an example of how you can do that. So forward passes through the model are relatively cheap, so we have a genetic variant of interest. We can do one allele and the other and compare, contrast the difference. And now the compare contrast opportunity is this really rich output of basically a full RNA-seq track prediction for all of the different training data sets that we worked with for each of the alleles. So there's a lot that you can try to learn from that. In this case, it's a pretty simple case of an eQTL. You just have greater expression across the entire transcript from the alternative allele. And you can see here, the top track is model predictions. The second track is us pulling the actual whole blood measurements from the GTEx project. So each of these will be a side-by-side comparison of model predictions with the actual data. So for this one, it's pretty interesting. So we want to better understand the genetic variant. The prediction's good overall, but what's actually happening there? I've talked about gradients. This is the other primary interpretation technique that we use, where we just go in and mutate every possible nucleotide within a certain window to all three alternative nucleotides. And then you can plot the height of the nucleotide in these visualizations as a function of the average difference between the reference and the alternative three alleles. This one I think is particularly interesting because in the alternative allele, there's these three motifs that pop out to form this enhancer element. The genetic variant is only hitting the middle one, CEBP, again, coming up. The other two are there in the reference. You can kind of, if you zoom in there, see them trying to pop out, but there's just not enough happening. It's not enough strength of motif power to pull this into becoming a full-on enhancer. And so it takes that third motif to really pull those motifs out, those other alternative motifs out of the sequence. So that I think is an interesting example from which one might generalize some principles. On the right, I'm just showing you our genome-wide accuracy of being able to pick out eQTLs. So the first way we set this up as a benchmark is we take the set of GTEx eQTLs that have been successfully fine mapped down to very high causal probability. These are cases where the linkage disequilibrium in the region, you just got lucky enough where you can pretty much pick out the causal variant. So if we use that as our positive set, and then we choose a negative set where we control for some basic properties of those genetic variants, then we can ask the model to be able to classify the two, and we can compare to the informer and see that the performance is much better. Each of those dots is one of the GTEx tissues. And at the bottom, you see how this changes as a function of distance, and it's pretty consistent in the boost over informer. So that's nice. So a very ambitious goal for this type of work would be to have these models look at a large cohort of humans and be able to impute gene expression experiments. If I look at this room of people, if I could impute your RNA-seq measurement in a very specific region of your brain, that would be phenomenally cool. If it was something that was paired with a medical database, then I could look for correlations between this imputed expression and different phenotypes. And the imputed expression would ideally aggregate all of the eQTLs, including a couple things that are pretty hard for these models to do right now. One of which is think about rare variants, and second of which is think about genetic interactions. So this would be wonderful. And some folks have forged down this path and realized that the state of the art is unsatisfying, that the informer model is not particularly good at this framing of the problem of predicting expression across a population relative to a simple regression model that's built on top of the eQTLs. So we want to get better at this. I'm going to say right now, it's not there yet. But I think a simplification of this problem that will help us get there is this scatter plot I have on the left, which is basically we need to be able to accurately predict the coefficients of the eQTLs, not just figure out what is and isn't an eQTL, but actually get the coefficient right. Because this framework is often called a transcriptome-wide association study. The models that we use for these are seeing dozens and dozens of examples in most cases of both alleles. So they are getting the answer right. There's no way around it. We have to be very close to that in order to allow for these benefits of rare variants and interactions to actually provide any benefit at all. So on the right, you can see the whole blood. This is where we're at. There's still a lot of stuff happening in the off-diagonal quadrants. These are like wrong answers, cases where a motif is recognized but misinterpreted, cases where the nucleotides flanking a motif, their contribution is misinterpreted. So we're going to keep trying to work on this, but I think it's hard. Okay, so everything I've talked to you about so far are things that the informer model could do, but I've tried to convince you that this is a kind of new and interesting approach. So let's dig into some other aspects of post-transcriptional regulation that are now accessible through these models. The first of which, and this was something I didn't know a lot about before, the model is incredibly good at picking out the end of the transcripts. I wasn't sure if this is going to be hard or not. Naively, I just hadn't read up much on polydenylation and transcriptional termination, but the model's quite good at figuring out where transcripts end. You can kind of see this qualitatively from the example. And then in the scatter plots here, we're computing a statistic for genes where there's two reasonable polydenylation sites and looking at the ratio of the coverage for suggesting one versus the other is the primary polydenylation site. And the accuracy for the model predictions versus the real data are very good. Again, we can come in with these interpretation techniques, showing you three different views into the sequence there. And the first thing that pops up way above everything else is this core polydenylation hexamer. But we can extract some other stuff too. And again, using the TF-MODISCO pipeline to cluster across lots of examples, we find that there's a number of known regulators of polydenylation that are identified by the model. So from these RNA-seq experiments done in populations, eQTLs are probably the thing people talk about the most, but you can also call various types of transcript structure modifying QTLs as well. So you can call polyadenylation QTLs from this data. These are cases where you can kind of see in the examples, there's not necessarily a change in the gene's expression. Sometimes there is, sometimes there isn't. But what's more prominent is a change in the usage of one polyadenylation site versus another. So you can kind of see these subtle shifts between the reference and alternative in red and blue in the second row there, representing the real data. So we wanted to see how well the model could predict these things when we introduce that genetic variant into the sequence. And the first row is showing you qualitatively, this looks pretty good. These two examples, we can go in and use mutagenesis style interpretation techniques to try to understand what's happening. If it breaks the core polyadenylation hexamer, that's too easy. So here's a couple examples where more subtle things are happening. In the left case, we have a sort of gain of function of a motif that's probably, yeah, I'm not sure exactly which one that is. Maybe it's the TGT CFIM factor. So the reference doesn't have it, the alternative does. That strengthens this polyadenylation site, which is why you see more aligned reads stacking up right there where the transcript will now more likely end. On the right, you have a case where the alternative allele is breaking one of these motifs that enhances the polyadenylation. And so then you see the reference coverage leading up to that polyadenylation site is higher, and the alternative, after you break it, the alternative coverage comes down, and there's a little more coverage floating out there for this downstream, more distal polyadenylation site. So again, we can play this game of, if we focus on the fine-mapped polyadenylation QTLs, where we're pretty sure we know the causal variant, and then choose a negative set to control for some basic properties like the gene's expression and distance to a core polyadenylation hexamer. Then on the bottom, you're seeing a variety of views into our current metrics for being able to predict these things. So it's pretty good. It's better as you get closer to the polyadenylation site. And the lead author on this work, Johannes Linder, before having worked on Borzoi, worked on a method called apparent and apparent 2, where he's fitting a polyadenylation sequence model from some reporter assay data, which is arguably the state of the art for this problem. And we're able to see boosted performance versus apparent 2, which is good. Okay, we've also got splicing here. So, I mentioned before the Splice AI program. There's Splice AI. There's another one that is a newer version called Pangolin that we like. It has an open source license. Splice AI does not. Just like to point that out as someone who works for a company and releases all of their software open source. Pangolin is also nice in that it makes some amount of tissue-specific predictions, which is the real thing that could be offered here by the Borzoi model. Is it can make different predictions for all sorts of different cell types. So I'm going to come right out and say now, the model, disappointingly, did not learn to predict tissue-specific alternative splicing very well. I would say this is the primary focus of my life right now, to try to figure out how to get this to work. And if you have any ideas after having seen the presentation, please come talk to me. So nevertheless, it's very good at predicting the presence of exons that are, shared across all sorts of different cell types. And a lot of splicing QTLs are, just stuff that's going to change across a bunch of cell types. So we can nevertheless set up an evaluation for splicing QTLs and see that, again, by playing this game of taking the fine-mapped splicing QTLs versus a negative set, that we can achieve compelling accuracy. One thing we found here is, Pangolin is very good at what it does. And despite the fact that this model is kind of looking at a broader context and looking at the aligned read coverage, it's a richer amount of information coming through in the training data set. It isn't a huge win over Pangolin. So this will be an area of future research. Interestingly, though, in each case, we tried the combination of the two methods, effectively creating an ensemble from the two different independent modeling exercises. And in this case, I would have thought there wouldn't be a huge benefit in that because these models are trained in a very analogous way, but there really was. So if you want to think about splicing QTLs, arguably the state of the art is to run both of these programs and use the result of that. So I argued that that there's interactions between regulatory layers and that there's some value in modeling these things together. So I need to show you at least one example of that. This is a case where when you have polydenylation information in an intron, there becomes a competition between the two machineries. The splicing machinery, which is trying to remove that intron, and now the polydenylation site doesn't matter anymore. It's gone. It's not going to terminate the transcript. Versus if the polydenylation machinery gets there first, then the transcript terminates. The splicing machinery can't do its thing. So there's this, it's a race, basically. And the strength of the sequence information can influence who wins that race, which of these two machineries. So there's an example here that we found, and there's a bunch of examples in the data sets. On the right, we're showing our ability to predict them, but the specific example will really give you a flavor. So this is basically a case where a mutation near the acceptor site of the splice junction is strengthened, which causes a weakening of the polydenylation. So it's more likely to pull out this intron, and that's why you see, maybe I've got this backward. These ones are relatively complicated, but hopefully you get the flavor. I could come back to this, but I want to make sure I leave time for questions. Okay, so all of this adds up just to kind of zoom out to a research program that we're pushing forward, which I call functionally informed statistical genetics. We want to be able to fit these models, have them try to understand everything there is to know about regulatory sequences, and then put them to work in the framework of statistical genetics for large human cohorts to be able to find map associations, identify target genes, and determine the specific cell types that we care most about. So we also did some analyses where we're looking at kind of general things about genetic variance and fitness. So to summarize, I introduced a framework here for thinking about genetic associations using these machine learning models of sequence to function. I introduced the Borzoi model, which predicts RNA-seq and combines multiple layers of regulation and predicts all these different kinds of QTLs quite well. I want to acknowledge the team at Calico that worked on this. Johannes Linder in the middle there really was a driver of creating a lot of these wonderful analyses and figures that I showed you today, as well as other team members, Divyanshi and Han. And you can check out the preprint describing more of the details of this work here. And I just want to quickly note that we're currently hiring into my group at Calico. So if this type of work excites you, then scan this or check my Twitter or go to the Calico website, careers website. Thank you.
We have lots of time for questions. Should I set the mic over there or is the room? So those who have questions, if you can come up and line up over here. Is that the idea, sir? If you want to put the microphone there, or
either that, or if it's easier, even if you wouldn't mind repeating the question, just so that it goes
Sure.
through the mic.
Okay.
Sure.
So when people are lining up, I'll start with the question. So what do you think about enhancer synergy and synergistic interactions between enhancers that your model may or may not be getting, right? If an enhancer on its own doesn't have any impact, and the another one doesn't have an impact either, but then them together have an impact. So what do you think about informer or Borzoi kind of approaches, and the ability to capture them, given that the validation data sets for them is going to be also extremely limited.
Repeat that.
I'm glad.
Okay. Yes.
Just the practice.
So the question is, I think that there's a lot of folks studying topics related to interactions between enhancers, different cases of compensation or synergy, and to what degree can the model learn these things. So I categorize these exercises as things that we would really love to do, and I've kind of paused because I don't know that we have the right validation data to actually assess whether the model's learned anything reasonable at this point. So if there was particular data sets, I know there's papers where people have gone deep on individual genes, and maybe that's sufficient, but always feel a little more comfortable when there's more genes in the mix. So, the model has the capability of learning something like this. And if we as biologists believe that it's happening, then whether the model has learned it really just comes down to, are there enough training data examples where it can identify that as a phenomenon and then start to generalize it? And I would hesitate to claim that that's happening at this point. Although, again, I do think it would be really interesting to kind of do all sorts of crazy in silico experimentation along these lines, and at least try to figure out what is happening inside the model, and maybe that helps guide some experimentation. So, interested in that topic.
Cool. So for predicting the EQTL coefficients, I was wondering how could that be better? So essentially, is it a problem of like the variant frequency, or sort of what other data do you think would help predict it better?
So as a simple research mind, I default to just getting more training data as the typical way to make my machine learning model better. So in that case, the options are the following. We're already training on human and mouse data, and we've done some work to show that adding the mouse data actually does help. So there's lots of other species that could be a source of more training data. Other folks, when you read those preprints, I think I can guess that they believe the solution is to train on genetic variation in humans and have measurements that are specific to individual genomes. And it's kind of a big, it's hard to say how big of a problem this is. The way we train these models is we pretend HG38 is the genome underlying all of these different data sets. And especially in the case of all the ENCODE data, there's a bunch of cell lines, there's a bunch of weirdnesses. That's an approximation that is kind of necessary to do a large-scale multitask training, where like you want to process the sequence once and then get gradient feedback from all these different tracks all together. But somebody will crack doing this efficiently, and we'll get the answer soon, whether training on genetic variation. So we can take all this GTEx data and we can basically bring through individuals' genomes and then predict their specific tracks. And we'll see. It's not a lot of new training data because ultimately humans don't vary that much, but it's focused on these EQTLs where we're currently getting the wrong answer. So it very well could be that even if those models are not learning a whole lot more in terms of generalizable principles, just by looking at the EQTLs, it may be a nice kind of interpolation of what the TWAS models are already doing, which is looking at both of the alleles in these models. But it's not going to solve if it doesn't generalize, it's not going to solve the problem of here's one experiment from one really rare cell type. I didn't do an EQTL study. I only have one sample, and I want you to impute it in the whole population. That's my dream, and doing this genetic variant training may not solve that. Great question, thanks.
Where do you think we're at in terms of the rate of return on growing model sizes? Is it still the case, besides making your training data bigger, is a bigger model still always better, or how much better?
It's this doesn't work like it does in the. So okay, question was, should we make the models bigger? Is that going to be a path to better accuracy? Okay, couple things. So one, we did shrink the model a little bit relative to Informer because we wanted to be able to capture other aspects. We wanted to make the sequences bigger. We wanted to predict a finer resolution. We're already maxing out the 40 gigs available on our A100 GPUs. So we had to forego a few things from the Informer, which probably would boost the accuracy. So I could come back to this exact problem set with a bigger model and achieve better accuracy. Genetic variant effect predictions at a UK Biobank scale are already very hard with the computational expense with this. So that's not high on my list of things to do because I kind of want to, predict every rare variant in UK Biobank. So there's a little left on the table, but I think it's quite different than how people talk about scale with the language models. With the language models, you have effectively an infinite amount of data. I mean, it's not really, but from the perspective of training these models, you can like deciding whether or not to look at the data a second time is a big decision they make. They could just process new language data almost forever. And that's not the situation we're in. We're data limited. So it's not necessarily the case that just making the models bigger will be better.
Given CBP's known methylation sensitivity, do you think that might be driving your blood versus liver tissue specificity that you observed? And could you use your models to actually delve into that and incorporate, say, methylation data with respect to transcription factor binding?
Yes. Question is, with CBP, there's a known role of DNA methylation, and what are some ways that we could bring methylation into the modeling framework? So we're not currently doing any modeling of DNA methylation, mostly because it doesn't fit the framework of all of the other data sets where like an aligned read coverage track is the primary signal information. It's more subtle than that. If you used arrays, then it's only specific sites that are measured. If you used whole genome bisulfite, then it's a ratio and you want to model the count statistics, and it's a lot more complicated. I would be both interested in trying it as one of the output of the models to just see how well we can predict it. Other people have done that. You can predict it reasonably well. But I think the idea of using it as input is intriguing. I tend to default to I only want the sequence as the input, because if I want to score genetic variants, then I don't want to use a source of experimental data as an input that could be modulated by that genetic variant too. But in the case of DNA methylation, it's the most stable epigenetic mark. It could be that it was laid down a long time ago in the history of that cell, such that the ability to predict it out of the sequence has weakened over time. So I think there's some interesting questions you could ask there for sure.
Thanks.